Hey guys, welcome to my video. In this video, I'm going to discuss all the questions in SPM Biology 2019 past year questions. Alright, so let's get started. Look at the question 1. Question 1. Diagram 1 shows a structure in one of the systems in the human body. Now, what is this? This is actually the structures of villus. And villus is actually the part of the small intestines. What is the function of small intestines? Digestion and nutrient absorptions. So from here, we can get the answer. This villus is actually come from the digestive system. Question 2. Diagram 2 shows a Paramecium species that live in the fresh water. This is the Paramecium. Okay, so let's bring the explanation here. Let's say this is the Paramecium. And from the questions, we know that this Paramecium is living in the fresh water. And we have the structure Y in the Paramecium. Now, first of all, we need to know what is the structure Y. Structure Y is actually the contractile vacuole. In one Paramecium, supposedly they will have two contractile vacuole. Now, what is the functions of contractile vacuole? Contractile vacuole is a vacuole that helps the Paramecium to control the water content in the body. So we call it as osmoregulation. Osmo means water, regulation means regulate. Now, why this contractile vacuole need to control the water content in Paramecium? As I mentioned earlier, this Paramecium is living in the fresh water. If you compare the water content in the water and the water content in the soil, the water concentration in the fresh water will be higher compared to the water concentration in the soil. In this case, we can say that fresh water is hypotonic compared to the soil, which is hypertonic. The fresh water will keep on diffusing into the Paramecium through osmosis. Now, try to imagine, if there are a lot of water moving into the Paramecium will eventually burst. So this contractile vacuole, it will contract to remove or pumps out all the water to prevent the Paramecium from bursting. Now, look at the questions. What will happen to the Paramecium species if structure Y does not function? So let's say structure Y can no longer function. It means that they cannot pump out the water. Water will keep on moving into the Paramecium, and then eventually this Paramecium will burst. So this answer should be A. Now, in biology, there's another word to replace burst, which is lysis. So let's say you see these words in the SPM in the future, please remember that they are having the same meaning. Question 3. Diagram 3 shows a bar chart of the density of organelle X in two types of cell. Organelle X. Let's look at the x-axis, the type of cell. Here we have the liver cell, the flat muscle cell. The y-axis we have the density of organelle X. By looking at this graph, we understand that the liver cell has high density of organelle X. But for the flat muscle cell, it has the low density of organelle X. What is the functions of X? Now, in this kind of questions, normally students, when they see the flat muscle cell, they understand that the flat muscle cell will contract and relax to produce the movement. To contract and relax, energy is needed. What is the organelle that can produce energy? The mitochondrion. Am I right? So they will think that the answer should be C, produce energy in the form of ATP, which is wrong. Please bear in mind that flat muscles and the liver cell, both of them, they need a lot of energy. So in this case, if organelle X is mitochondrion, then the graph should be looking like this. They have almost the same density of organelle X. If organelle X is not the mitochondrion, then what will it be? Let's check out the functions of liver cell first. Some of the important functions of liver are metabolysis, carbohydrates, and detoxification of drugs and poison in our body. So to carry out these two functions, they need a lot of enzymes. And this enzyme need to be transported out from the cell. And what is the organelle that can transport the synthesized enzyme? The Golgi apparatus. So this answer should be B. Organelle X that transport the synthesized enzyme out from the cell. So let's check out other options here. Produce the spindle fibers. Normally spindle fibers is for cell division. Liver cell and flat muscle cell, they are not active in cell division. That's why A will not be the answer. Now let's look at option D. Option D, secretes mucus to protect the mucous membrane. Liver cell and flat muscle cell, they are not the cell which is able to produce the mucus. So answer D is wrong as well. Okay, so let's check out another questions. Diagram 4 shows the movement of the substances across the plasma membrane. So what you can see here is there are some substances outside the cell trying to move across the plasma membrane to the inside of the cell. Check out the molecules outside the cell. We have 3 of them here. And inside the cell, we have 5 of them here. Now, in this case, this substance is moving from the low concentration area to the high concentration area with the help of the ATP. So from here, I'm sure this transport is known as the active transport. So now look at the questions. Which of the following statements explains the movement of substances in diagram 4? Movement of substances going down the concentration gradient. Now, what is the meaning of going down the concentration gradient? Going down the concentration gradient means the substance move from a high concentration area to the low concentration area. So this is the meaning of going down the concentration gradient. As we mentioned just now, the active transport is substance moving from a low concentration area to a high concentration area. So A is not the answer. Let's continue with B. Movement of substances against the concentration gradient. So this is correct. Substance against the concentration gradient, which means that the substance is moving from a low concentration area to a high concentration area. This is the meaning, against the concentration gradient. But you look at the last part of this sentence, with the help of the pore protein. So remember, pore protein, it has another name called channel protein. If you look back to the diagram above, you will notice that this protein will change the shape, which can fit the substance. So if they can change the shape and this protein is using ATP to transport the substance, I'm sure this is the carrier protein. Okay, so if the option is saying with the help of pore protein, then question B is wrong as well. Now look at C. Movement of concentration from the region low concentration to high concentration. This is the active transport. Now with the help of carrier protein, yes, all correct. So the answer supposedly is C. Now look at option D. Option D, movement of substances from the region of high concentration to a low concentration. Now high concentration to low concentration, this one is the passive transport. So D is wrong. Diagram 5 shows clothes hung on the clothes line. So the clothes is made by silk. Surrounding temperature 45 degrees, which is considered is quite hot, and relative humidity is very, very low, which means that the environment is dry. Now, what will happen to the clothes after 5 hours? The answer for this question is clumper. Why? Because silk is made by protein. And if you zoom inside to the protein structure, you will notice that this structure can maintain the shape because it has some bond in between the structures. Let's say you are exposing the protein to the hot and dry environment, then this bond will be affected and eventually the silk will be crumpled together. So this is the reason. Alright, so let's continue. Question 9. Diagram 6 shows a type of human blood cell. This is the red blood cell. Which of the following protein structures can be found in the blood cell? You cannot find the answer for this question from your KSSM textbook because this part it has been taken out by the government, but no harm to know more, right? So let's continue. D is the quaternary protein structure. So the hemoglobin of the red blood cell is actually made by the quaternary protein structure. So this answer is D. And how about C? C is the tertiary protein structure. And I can give you an example. The proteins such as the enzyme. Yeah, they are made up of tertiary protein structure. For A is a primary protein structure. And for B is the secondary protein structure. Let's continue with question 7. Diagram 7 shows an experiment to study the reactions of enzyme at the temperature of 37 degrees Celsius. So inside here, we have the lipase solution. Now, what is the functions of lipase? Lipase, it digests the fat into the fatty acid plus glycerol. So this chemical reaction is done by the lipase enzyme. Now let's continue. We can see this lipase solution is mixing with the full cream milk. Full cream milk is full of fat inside. So supposedly the lipase enzyme can digest the fat in the full cream milk. Look at the question. What are the results after 15 minutes? We have the product for the first column and the pH value for the second column. The result after 15 minutes, I predict the lipase here will digest the fat in the full cream milks and turn them into the fatty acid and glycerol. Since we only have lipase enzyme in the solution, so no amino acid can be produced. I remove A and B, left C and D, which is the fatty acid. How about the pH value? Since fatty acid is a type of acid, and this acid will lower down the pH value of the solution, then the answer should be C. Okay, look at question 8. Diagram 8 shows a process that occurs in human digestive system. Which of the following is Y? 3H2O plus, what is that? The triglyceride. This molecule is known as the triglyceride. These reactions involve 3 molecules of H2O, which means that this is the reaction, we call it as a hydrolysis. Hydrolysis is a process that a big molecule is broken down into a small molecule. So from here we can see that the triglyceride is being broken down into 2 molecules. This molecule is known as the glycerol. And supposedly Y will be the 3 fatty acids. So the answer for this question is B. Continue. Diagram 9 shows R phase in cell division. What is the next phase after R phase? Okay, the keywords here is next phase after R phase. We are going to find out what is R phase first. Now from here, we actually can notice something. Can you see the chromosome is having different colors? Why these 2 chromosomes are having different colors? Because these 2 chromosomes, they come from a process called crossing over. Now, crossing over only occurs in the prophase 1. Besides, the chromosome inside the cell is still in this shape, the ribbon shape. This cell just came across the separation during the anaphase 1. Now, let's look back to this diagram. So this diagram showing you one cell is trying to be separated into 2 cells. I believe that this R phase is the telophase because the chromosome, they are still in the ribbon shape. What is the next phase after the R phase telophase 1? The answer should be the prophase 2. So prophase 2 is just right behind the telophase 1. Question 10. I brought you a picture from a Google image and I'm going to use this picture from the Google image to explain these questions. Now, question 10 say that which of the following diagram of the cell division is matched correctly with its phase? Okay, so before we start, I want to go through the whole process of the meiosis 2 first. At first, you have 2 chromosomes here. And these chromosomes, they are not in the ribbon shape. But when it comes to the prophase 1, the DNA is replicated and they will form the ribbon shape of chromosome. Then eventually, they will be separated by the spindle fiber and the chromosome is still in the ribbon shape now. Even though come to the prophase 2, the chromosome is still in the ribbon shape. So once it's being separated by the spindle fiber, it will form a chromosome which is not in the ribbon shape now. Check these questions out. Options A, the chromosome is no longer in the ribbon shape, but the phase is prophase 2. Let's check out what is prophase 2. Prophase 2, chromosome still in the ribbon shape. So supposedly, A is not the answer. B, prophase 1. Prophase 1, from here we can see this is the process of crossing over. And remember that crossing over can only occur in prophase 1, which is exactly matched to its phase. So answer is B. How about C and D? C, metaphase 1. So during metaphase 1, all the chromosome will line up at the middle of the cell. And you can see the chromosome here is not lining up at the middle of the cell. So answer C is wrong. For options D, telophase 1, the chromosome, they are not in the ribbon shape. So this one is actually belongs to the telophase 2, which is this diagram. So option D is wrong.